Just very quickly, what is Gabather? Gabather stands for GABA therapeutics. I go into what GABA actually is. It's a neurotransmitter, very important one in the brain. The key thing is that we work in drug development and mental health, i.e. psychiatry and neurology. And second key message is that we actually develop our own small molecular weight drugs from bench to bedside. We do not license in and license out again. We synthesize our molecules and our drug candidates ourselves in our lab in Södertälje, in the AstraZeneca facilities actually. The status right now, we're going to talk about a lot of it today, is that we have developed a drug from the bench, from the lab tube to a clinical stage 1. Actually, you can also define as stage 2 because we're looking at drug effects. We have a very interesting patent series, and we do have a lot of compounds in the pipeline we can work with. But I'm going to focus on our clinical candidate today, which is the one that's of most interest. Right now, we are doing a rights issue to get a runway to finish our project, and I think I can convince you today that this is actually an interesting and very important project. It actually originates, and this is now many years ago, from the University of Lund and the University of Copenhagen. Actually, I was involved at both the work at the Lund University and Copenhagen, and so was the entire team actually. And we are a very, very small company. In fact, all three management drivers are here in the audience today. But it's very, 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 very experienced team, so we can actually do quite a lot with a very few key resources. I'm going to talk a lot about mental health today, which is, of course, a topic that is on and off the charts. I think it's always actual. I think it's always relevant. And if you look at any all of these diseases are familiar to anybody in this audience, and you can discuss about the numbers, if it's 1 in 4, 1 in 5, or 1 in 6. Depends on which statistic you look at. It's a big societal problem. It is also a problem that is a lot of taboo. And nobody really wants to speak about being very, very depressed and not be able to show to work. The socioeconomic impact is also huge, and I don't want to make big numbers figure out of it. It's enormous. There's a lack, actually, of innovative medicines. There are medicines around, but there are no new ones, and there's a lot of room for improvement. And that is what we mean by the last bullet points here is there is a very, very large unmet medical need. An unmet medical need is defined as the therapies that are better than what we have now, either lesser side effects or better efficacy. I cannot avoid being a little bit technical, and GABA is the name for a neurotransmitter, which together with the neurotransmitter glutamate, these are the two main drivers in our brain. The GABA, to put it very automobilistic, is the brake in our brain. It inhibits the brain activity, it slows us down, and glutamate excites us, speeds us up. So it is the balance between the GABA and the glutamate system that regulates our brain activity to a large extent. This is not to say that there are a lot of other neurotransmitters which are as important, like dopamine, adrenaline, and so on. We focus on GABA. We've developed our molecules targeting this particular receptor, this neurotransmitter in the brain. This is what we modulate. And this is what we have done in milestones in our project. This is very much a focus on the clinical aspect. And the reason for that is in any drug development, if you are in this business, the step between preclinical, what you can show in a laboratory or in an animal model, and in a human being is the biggest step you can have as any company in drug development. And we've done that step a couple of years ago. And I can tell you that it's the most sweaty time in any project when you're sitting beside your phone or your email and you're waiting for the first news when your developed drug has come into humans for the first time. We've gone beyond that, and I'll show you later. We've gone into the third clinical trials in man, and we have shown that this drug is very safe and very well tolerated, which is the biggest goal to achieve in any development, actually. So we've done both studies called SAD, strange acronym, stands actually not for anything that's depressing, but single ascending dose. you give to healthy volunteers, and we've also completed a MAD study. There you increase the doses, you give them multiple ways. And all these studies are just to check that the drug actually is safe and well tolerated. And that has our clinical candidate GT002 passed with flying colors so far. And we'll be moving ahead, and the reason I'm speaking today is we're doing a target engagement study where we look at the effect of the drug. Does it go into the CNS, and does it modulate the activity in the brain? And it does. So the next step would be the last clinical trial, phase 1, and then we will plan and go forward. Once we have our baseline, we have our basis very, very solid, we know what this drug does, we can start to look at treatment of patients. And what kind of treatment, what kind of patients we are? We have very good preclinical evidence that this compound actually has a positive effect on cognition, as in our case, memory and learning. And the interesting thing is, if you look at the top row here, this is just a bunch, by no ways complete, of neuropsychiatric disorders. And the interesting thing is, if you look at the symptoms that we've listed in the columns, you can see that many have, of course, very specific symptoms, but in most of them, you have some form of deficit in cognition, i.e. in learning or memory. And this is something that all of us know. If we are very, very stressed, we know that we forget things, we rush things, we don't know where we put the car keys or park the car in the first place. So if we can target cognition, and that is what we want to do with our drug, we can actually all of a sudden become interesting as a therapy for all sorts of neuropsychiatric disorders if we attack that particular symptom. So it's not the ambition to be able to cure disease, but it's certainly an ambition to cure a very specific symptom. And if you think of a disease like, very much in the press lately, Alzheimer's disease, Alzheimer's dementia, where deficits in cognition is the absolute key symptomatology. And if we can go and help or develop a therapy that could be at least helpful in that particular symptom in Alzheimer's disease, I think we've had a very, very big step. This is, of course, an ambition of us. And I don't want to turn this into some sort of science seminar, but this is just a very simple way of trying to explain the way we experience the world with cognitive processes, both with reason and emotion. They, of course, affect each other. So the experience of the world, if that cognitive deficit is deficient, the reaction to our world will be different because we don't really understand it anymore. We can't follow it anymore. It becomes diffuse, like in this analogy. And of course, our hope and our goal and our vision is that we can develop a drug, in our case, our candidate, GT002, that can restore this cognitive processes to such a way that you can react to the environment, and your experience becomes that you can interact with society again. One of the biggest effects of all the symptoms I've showed you before is social isolation, of course, which is due to deficits in the cognitive processes. So it's a very high ambition, of course. How do we know that we actually do something positive in the brain? We have chosen one biomarker, one way of measuring what we actually do with our candidate drug, and this is EEG. Stands for electroencephalography, i.e. we measure basically the electrical activity in the brain. The brain is an electrical apparatus. And the interesting thing is that if we measure this and take this as our sensor, so to speak, we can actually look at patterns that are specific and relevant to the different diseases, and look how do they modulate the EEG, how is the EEG modulated in these diseases, and then see how our drug, by looking what effect does it have on the EEG, can modulate and perhaps be used in these diseases. Again, this can be very varied one. The measurement that's common to all of them is that you measure the electrical activity in the brain, which is what is deficient in these diseases, neuropsychiatry. So you can use it as a biomarker in healthy volunteers. That's what we are doing right now. And you can also, of course, once you have this pattern, and we have a presentation after the study, and I think we have some actually very nice idea how we can use these patterns to go for a very directed way to look if our drugs work on a specific disease, and then we can test them. Eventually, our plan is then to go into patients, because the proof comes with the pudding. And just another very small side remark. If you think about cognition and cognitive defects, and then you're actually also going into areas like healthy aging, because we all know that during aging process, memory, for example, in learning actually becomes slower. And this is just, I can go skip this over. The key thing is that we are at phase 1, going into phase 2, which we actually look at effects in patients, of course. Phase 3 and phase 4, this big pharma terminology, is nothing that we want to approach right now, because we know you need a large number of people tested, patients tested, and that can make it very costly in a very long time. So we are looking at, with the war chest that we have built up of a very safe phase 1 drug, we are looking now at the effects on the borderline here, because we're looking at what effect does the drug actually have now that we know that it's safe on the brain by measuring EEG. So we are somewhere in the borderline here with our project. And this is my idea. We have pushed this one project, as you can see, through all the phases from discovery through preclinical into the clinic here, very much based on one drug, and that is because, very simple, we have to show that our drugs and the molecules that we synthesize and have patented have a potential in the clinic. Nobody wants to fund 5, 6, 7, 10 years of preclinical research. So we have pushed our lead candidate very hard into the clinic, and now, as I mentioned before, there's a number of indications, i.e. diseases that we can try to treat, but we not forget that we do have a pipeline of drugs that we can synthesize in our own lab. Again, this is a bench to bedside process, which we've done to the bedside only for GT002, but we can do the same for others. We have 2 patent groups, actually. So where do we stand? What have we learned of this? We have an orally available, might sound trivial, it is not trivial, a well-tolerated, orally available drug is very useful with the clinicians, so that it's well-tolerated and safe. We've upscaled the clinical material, so we have a lot of material we can use, and which is actually a big advantage for people that want to work together with us, because there are not that many companies that have a candidate clinical trial material on the bench, on the shelf, in order to be tested. And we've actually patented and developed the first form of this product. There are others to come later. And that is just the snapshot of where we stand right now. The patents are already mentioned. We have, I think, a very straightforward patent strategy. We've covered our compound, the molecules. We've patented how to use them, and we have patented also in what diseases we can actually try to use them, in what indications. And right now, we are finishing the 3rd and last phase 1 clinical trial and going into phase 2 towards the end of the year. And I will leave you with this too. We already talked about the imaging techniques before, so that as we speak, there are people being treated with our candidate drug. They are measured using EEG to look at activity, and they also measure with fMRI to look where in the brain is the activity and which relevant brain areas. Some are more relevant than others, depending on which disease. And it's a double-blind study, so neither the investigator nor the patient knows what they are getting. And it's a crossover disease because we're comparing with current medicine. So we can actually test the quality of this clinical trial by ourselves. And we are waiting for the finalization of all the healthy volunteers by the summer this year. So then finally, what's in this kind of projects? We are, of course, looking for licensing opportunities. We are too small to run this onto the market. We are looking for partners, and there will be medium and big-sized pharma anywhere in the world. We have a big network. We've talked to most of them. We are on the radar of most of the business development company as potential partners, both for clinical trial, but also for licensing of the compounds. And there are some examples of some of these deals that are made with pharma industry. And of course, the most relevant one or the most interesting one in our particular field is the deal that was made with Biogen and Sage Therapeutics. And this is relevant because not only is this an enormous volume that actually the value of this deal, but it was a molecule that targets the same receptor system as we do, but it's a completely different molecular structure. And I venture to say that our molecules actually have a structural advantage and a formulation advantage over those. But it is, of course, the benchmark. What can you get on this kind of projects? And I leave you with this very last slide. And all this I've already talked about before, and you can read it by yourself. But one last thing I want to mention is that we've been contacted quite a lot lately by groups, networks, people that are working with effects of long-term COVID, because as acute COVID has disappeared out of general knowledge or interest, there are, of course, people suffering from long-term COVID effects, which affect, to a large extent, memory, concentration, i.e. cognition. So people are asking us, do you have any drug we could test, for example, in COVID-19 patients in the future? So I think there's something very interesting, something we had absolutely no idea when we started our project in Gabather. So I think this is my last slide, and I leave you with it. And we are a very, very small team, both myself, Stefan and Christine. We are here today. If you're interested in this kind of projects, and come out and see us, we have some, I think, very interesting ideas how to make this drug, how to test the drugs, and what principle we actually are looking at. And if you take your time and take a look, it makes a lot of sense, and I think it's very, very promising. And you know, to already anticipate your question, how do you guys think that you can do something that Big Pharma has been struggling for the last many, many years? And I will tell you that the reason why I think we can succeed is actually this team. I think it's the quality of the people that can make a project win. And I think with that, I leave you. I thank you.